Welcome to today's webinar. USP 51 Antimicrobial Effectiveness Testing and USP 71 Sterility Testing is presented by Brian Kelly, Director of Business Development for ARL Biopharma. Brian has the overall responsibility for customer service, marketing, and sales departments. He received his BS in biology from Oklahoma State University. Brian's professional background includes 10 years of research, development, and project management in the biotech industry at Genzyme. Today's webinar is accredited through ACPE and is worth 1 contact hour. CE credit is applied following completion of an evaluation link found on the last slide in the presentation handout. You can find the handout on the lower portion of the GoToWebinar toolbox under Handouts. We are recording this session and will include the recording and evaluation link in a follow-up email 1 hour after today's live session. Questions may be submitted throughout the webinar using the GoToWebinar toolbox on the right side of your screen under the Questions tab. We will answer the questions submitted following the presentation. If we do not get to your question today, a list of questions and answers will be emailed to all attendees following the webinar. Thank you for joining today's session.
All right. Thank you, Amy, and thank you, everyone, for your time today as we talk about 2 tests that may be really important for your BUD assignments per the new 795 and 797. Again, reminder, official date of November 1, 2023. Okay. No conflict of interest to disclose. This is an educational course here worth 1 contact hour. Okay, so we're first going to talk about USP 51, but before we get into the 51 tests, I'm going to talk a little bit about what antimicrobial preservatives are, a little bit involved in the selection of which one is appropriate for that particular compound preparation, and factors that may affect that may reduce the effectiveness of the antimicrobial preservative. And then we'll finish off talking about sterility testing per the new 797. So if we're going to talk about testing, a good place to start is USP 1163. It is titled Quality Assurance in Pharmaceutical Compounding. That chapter says that compounders should know, should have some basic understanding of the tests, including, why to test, why it's important, when to test, what to test, how the test is conducted, what the results mean, what the results do not mean, and how to proceed should the test result come back as out of specification. Okay. So for a result to come back out of specification, you must first have a specification. Luckily for these 2 chapters, the specification is provided for you. So what are antimicrobial preservatives? They're functional excipients added to multi-dose containers of aqueous, non-sterile, and sterile, but with slightly different purposes because the microorganisms might be introduced at a different point. So for a sterile product, because the product is sterile, meaning it should be free from microorganisms, really worried about in those multi-dose products about microorganisms being inadvertently introduced during the repeated withdrawal of individual doses. So really concerned about what happens with that product after it leaves the pharmacy. For non-sterile products, because they are not made using aseptic technique and things, worried about microorganisms that might be introduced before compounding, meaning from the starting materials, worried about microorganisms that might be introduced during the compounding process, so from the compounding environment, from the personnel involved, from the equipment involved in the compounding process, and certainly again after the compound preparation is in the patient's hands. So of note here, the use of antimicrobial preservatives is not a substitute for good compounding practices. certainly not a good substitute for proper aseptic technique or terminal sterilization. So hopefully as we finish up the 51 talk, you'll understand why I made that statement. So starting off here is a table that we pulled from a paper by Vu, Nguyen, and Kupik with a list of the common pharmaceutical preservatives. This table, this is just the first part of the table, but the table includes information on what type of formulation those preservatives are commonly used in. Real important on the appropriate concentration for those preservatives to be effective. And an area where we have seen problems is when the formulation is not at the appropriate pH for that preservative to be effective against those microorganisms listed in the right-hand column. So just looking down, benzyl alcohol is a real common preservative used in compound preparations. The second part of the table here includes like the methylparaben and propylparabens. Those are often used in combination in compound preparations. So you may hear me throughout the presentation use the term antimicrobial preservative system because there may be, in fact, more than one preservative present. But this is mostly provided here for future reference as you're developing formulations. So the antimicrobial effectiveness test, it's a challenge test. So going to challenge those antimicrobial preservatives of can they do their job of preventing proliferation or limiting microbial contamination for microorganisms that are purposely added into the preserved drug product. Maybe thinking, well, I know my preservative is at the appropriate concentration. I know my benzyl alcohol is at 0.9%, for example. Why do I need to run the test, or why is a potency test not appropriate? That's because there can be a whole host of interactions between the preservative and something else in the formulation, including the API. We looked at the pH and talked about that a little bit. That might not be appropriate. The concentration may not be where it was intended to be, right? So think about some of those bases that you start with, or if you're starting with bacteriostatic water or something, and if you're adding things to it, API or commercial product that's not preserved, you may be effectively reducing the amount of that preservative present to a point where it's no longer in a concentration to be effective. So the preservative can also potentially bind to the container closure system. So all of that could minimize the effectiveness of those preservatives, and so that's the purpose of this test. So looking at 795, of when this test is required, we're really only talking about one type of preparation here from Table 4. We're talking about the preserved aqueous dosage forms, and if you want to exceed that 35-day limit. Okay. So the chapter defines aqueous as a drug product that contains water activity greater than or equal to 0.6. We'll talk about that a little more coming up. There's an important footnote below the table here that I want to point out that says a shorter BUD must be assigned when the physical and chemical stability of the compound preparation is less than the BUD limit stated. So certainly something to be aware of there. Similar footnote on the sterile side. So the first step is know your water activity, right? If you're below that water activity level of 0.6, then you do not need to worry about 51, because that means there's not enough water available in the compound preparation to support microbial life, right? The chapter 795 provides some example formulations and their water activity. So if you find something that matches well, then you can use that water activity listed in the chapter. But another option would be to just run the test. It's a relatively inexpensive, fast test, and then you'll know the exact water activity on your compound preparation. So if it turns out that your water activity level is at or above that 0.6, then you do need to have USP 51 data on each formulation and each container closure system. Okay, I stated that as you have to have the data because the chapter does allow for literature references as long as the testing was performed by an FDA registered facility or it was published in a peer-reviewed journal. The testing must be performed on the same formulation and the same container closure materials of composition. So again, concerned about those interactions between the preservative and the formulation, or the preservative and the container closure system, or both really. So you need to make sure those are exact matches if you're going to utilize data from a literature search. So switching gears over to the sterile side, 51 is required on all aqueous multi-dose compounds sterile preparations. Again, must have data on each formulation in each container closure system, and has the exact same rules there for the literature references being allowed. There's also bracketing is mentioned, which is a mechanism to reduce the amount of testing. This is a common strategy used in stability studies, right? So basically the idea being is, say you make 3 formulations where the 3 formulations are exactly the same, including the preservative concentration. The 3 formulations are exactly the same except for the concentration of the API. Then you just need to be able to show passing 51 results on the highest and lowest concentration of the API, and then any formulations that fall in the middle of those are also covered if you can show passing results for both the high and the low. So a couple of mechanisms USP built in to reduce the amount of testing required for 51 between the bracketing and the literature searches. So looking specifically about how this test is performed, so again, to challenge that antimicrobial preservative system, and how that is done is a fairly high concentration, 10 to the 5th to 10 to the 6th CFUs of each of these 5 microorganisms are added in directly into the preserved drug product. Those are done separately, so separate aliquots. And then there's some incubation period, and then counts are performed to see, are there still 10 to the 5th, 10 to the 6th range? Are there more or are there less? Over time, when you allow time for interactions between that preservative system and those purposely inoculated microorganisms. So looking specifically at the design of the study, at day 0 of the study, those 5 microorganisms again are added into separate aliquots of the preserved drug product, and then that combination of those purposely added microorganisms in the preserved drug product are incubated at 20 to 25 degrees Celsius. But also at that day 0, you need to make sure that addition of those microorganisms were at the 10 to the 5th, 10 to the 6th range. So you need to plate some control counts, right? Some controls. But the microorganisms are too small to see, so you have to not only plate them, but you have to incubate them for 7 days so that they're at a point where they're large enough to see, so that then you can count them. Okay, that's kind of a general theme that's going to occur throughout the study here. So at day 0, add the microorganisms into the drug product, plate some controls to make sure that it was actually at 10 to the 5th, 10 to the 6th. Then at day 7, for some drug products, that'll be the first time that you actually get into that combination of those purposely added microorganisms and the preserved drug product, and plate them out. And again, you have to plate them, incubate them for 7 days, count them. That same exercise is then performed at day 14, but that's for all drug products. So you get into that combination, plate it, incubate it, count it. and then perform that again at day 28. So if you notice, this is a 35 calendar day test. But on the front end, there may be media that needs to be purchased or made, any QC work on that media, purchasing of microorganisms, preparing them. I've failed to mention that there's a method suitability involved, which is a kind of formulation specific mini validation, if you will. But during method suitability is where it's determined of how to plate those samples, where it's a combination of the preserved drug product and those purposely added microorganisms. You need to be able to plate them in a fashion that they'll grow so you can see them, so you can count them. So there's method suitability. And then as you hopefully can see here, there's lots of data. I've got 5 microorganisms. I've got my control counts for each of them. I've got potentially day 7 reads. I always have day 14 reads and day 28 reads. So now I've got lots of data also. So at the end of the day, you should probably think about needing about 2 months to complete this test. Considering we're almost in April now, you're looking at June to have this data, and that's if the testing is can be started pretty much right away. So don't delay. Get started on this. November will be here before we know it. Okay. So now what do you do with all of this data that you have, where you know how many microorganisms were still around and still viable at these time points? Well, USP 51 divides all products into one of 4 types of categories. Just to simplify, I'll just say injections, topicals, orals, and acids. So I want to take a look at these, at the specifications that I mentioned earlier that 51 provides. So looking at category 1, which is again the injections, notice first there's different criteria for bacteria than for the yeast and molds, as those 5 different types of microorganisms that are added represent different various classes of microorganisms. So for the category 1 bacteria, at that day 7 count, there must be at least a 1 log reduction. This is NLTs, not less than. So not less than a 1 log reduction from that control count after 7 days of incubation with that combination of the preserved drug product and those microorganisms. There's also a criteria of not less than a 3 log reduction from the initial at day 14. So essentially maybe even a further kill off after 14 days of incubation. You could have a 3 log reduction at day 7 and still pass, but so anyway, at 14 days you have to have no less than a 3 log reduction, and then there can't be any increase in the number of CFUs at 28 days from 14 days. So the idea being is that they don't want to see like an initial kill off, and then where the microorganisms can recover and start proliferating again. So again, we're looking at the category 1 injections criteria for just bacteria. For the yeast and molds, just no increase from the initial at day 7, 14, or 28 days. So different criteria for the yeast and molds than the bacteria, but as you can see, there's lots of opportunities here for the calculations that have to be performed, and there's certainly scenarios where it could look like you're passing at day 7, day 14, and then it's not till day 28 that you find out that there's a problem. So do not assume that these are all going to pass. There's lots of data, 5 different organisms, all these different acceptance criteria. So just quickly here, we'll look at category 2, which is the topicals, just to make the point that these different category products have different acceptance criteria. So for the bacteria on category 2 products, there's actually there's no read at day 7 for these. So just after the 2 log reduction from the initial count at day 14, and then no increase from 14 to 28 days. Again, making sure that there's no kill off recovery, and then they start proliferating again. And then a very similar acceptance criteria for the yeast and molds for the topicals as for the injections. Just no increase from the initial at 14 or 28 days. No 7 days again. In the interest of time, we'll skip over the category 3 and 4 products, but this is just pulled right out of the chapter. Okay, so at the end of the day, may get a test result that says conforms or pass, or it does not conform or does not pass or fail. So if you get a test result that says that confirms or passes, still need to consider the chemical and physical stability for appropriate BUD assignment. If you do not pass or the test does not conform, you need to investigate the method and the results, right? There's lots of calculations there. Could question whether the method suitability was performed appropriately or not, but also may end up landing on potentially need to determine an appropriate preservative system for that preparation. Or think about, was my preservative at the concentration I intended it to be? Do I need to run a potency test? Should I run a potency test anyway? Because it'd be a real nice data packet to be able to show that I ran a potency test. My preservative is where I thought it was supposed to be, where it's intended to be, and it passes a 51. But taking into consideration that there is opportunities to not pass the test, if this is something where you're going to not pass the test and end up in formulation development, take that into consideration before you elect to proceed with a stability study, right? Because you may have to redo the whole stability study, including method development validation work, if you proceeded with that before you passed the 51 test, before the formulation passes the 51. So really think about order of operations there. Might save you a lot of time and money. So that concludes the 51 portion of the test. Moving on to the sterile side. So gone are the days of low, medium, and high risk compounding. That has been replaced with category 1, 2, and 3. The category is really based on the state of environmental control in the compounding area, the probability for microbial growth, and then the BUD, or the use period. So really look at those conditions and figure out where you are or where you want to be as far as which category you fall into. So looking at specifically the sterility testing requirements for the categories, sterility testing is not required for category 1. Their use periods are a maximum of 24 hours, just not enough time to perform a sterility test there. And then for category 2, if a sterility test is required, if you want to use one of the BUD limits assigned in Table 13. Performing those sterility testing adds a significant number of days onto your use period there, so certainly something to take into consideration. And then sterility testing is required for all category 3 CSPs. Mentioned earlier about the note about being concerned about physical and chemical stability. Note that it's also present here under Table 13. I'm certainly aware of some compound preparations that are not stable for as long as this table allows, particularly the ones if sterility testing was performed. So the chapter talks about sterility test methods. Says that USP 71 with membrane filtration is preferred. I would add another word to that, and I would say that closed membrane filtration is preferred. We'll talk about that. But not all drug products are filterable. So if it's not filterable, then direct inoculation, meaning the drug product is added directly into growth media, is the preferred alternative method. The 797 does allow for alternatives to these methods, but the method must be fully validated and method suitability has to be established. So Again, method suitability is a kind of a mini formulation-specific validation of the test procedure itself. In most cases, these alternative methods are going to be the rapid sterility test methods, which we'll talk a little bit about. But so it does have allowance for that, but it is setting a little bit of order of preference here on the sterility test methods. Note that those rapid sterility or the alternative test methods, the method itself must be validated and method suitability. If you choose to use one of the USP 71 methods, that method is already considered validated. You just need to perform that method suitability to be able to use those methods and consider them valid. So one of the things to consider, important note, is that sterility testing is really a lagging indicator that all of the other things you're doing with your facilities and your environmental monitoring and control, with all your staff training with appropriate garbing procedures and all those things, the sterility test is a lagging indicator that all of those other things you are doing are working the way they should. Right? So do not just depend on the sterility test. It's really more a confirmation that everything else you're doing is working appropriately. And that's because a passing sterility test only indicates that there were no contaminating microorganisms in the samples that were tested. It doesn't really speak to the whole rest of the batch. It does the best it can, but again, that's why all those other things you're doing are important, and the sterility test itself is really just a confirmation. So, why we're talking about this is because microbial contamination is not likely to be uniform across a batch. Really, for an entire batch of vials or bags or syringes or something to be contaminated, really going to have a gross contamination event, right? Like a stock solution being contaminated, that stock solution was then used in every individual dosage unit. But a momentary lapse in aseptic technique or something like that, that's not typically going to contaminate the entire batch. That will just contaminate certain certain number of articles, which is important when you collect the samples for sterility testing. Why you really want to be able to, you want to collect samples from throughout the batch. Right? Don't just grab the first 5 vials that are made or the last 5 vials that are made. You really want to sample from throughout the batch. And so there's really, because you can't sample the whole batch, we're not there technology-wise yet to be able to sample every vial or IV bag and still have drug product left for the customer. So you can only sample a portion of the batch, and with the portions of the batch that are sampled per USP 71, there's a good chance that if a low percentage of the batch is contaminated, that the sterility test is still going to pass. Okay, so this data says that if 0.1% of the batch is contaminated, there's still a 98% chance the test is going to pass. It's 1% of the batch is contaminated, there's still an 82% chance the sterility test is going to pass. So again, don't depend on the sterility test alone. Think about all the other things that need to occur to make sure that compound preparations are indeed sterile. A couple new rules from the new 797 is that the batch sizes have been capped at 250. So 503As can no longer make batch sizes of 1,000 or 500. Those are going to need to be split up into separate batches. The chapter also removed the allowance for early release with daily sterility reads. So that's no longer a thing. You do need to wait until you receive the final test results before those are being administered to a patient. If you are doing these today, just sort of a word of warning that daily sterility reads, that means that reads need to occur every day, Saturday, Sunday, 4th of July, Christmas. So if you're doing those and that's really a calculated risk, you need to make sure that all of that is happening to make sure that you're following the rules in the chapter there. The old chapter also used to have an exception for small batches that didn't need any testing on batches less than 25 units. That has been removed also. But for small batches, they have changed the sampling requirements for the sterility test. So for batches less than 40, the new rules in 797 say that you test 10% of the batch rounded up to the next whole number. Of note, there were not changes, or these changes are not documented in USP 71. They're only in 797, or that particular change. For batch sizes greater than or equal to 40, still follow Table 3 and 71 the way that's being done today. We're going to look at some examples of test sterility batch sampling. So say that the need was to be able to dispense 4 10 ml vials. Okay. Following the current 797, you go to Table 3. Table 3, you'd find the number of items in your batch, which is not more than 100, which then says you need to sample 10% or 4 containers, whichever is greater. So 4 is greater than 10% of this particular batch. So you have to make 8 vials so that there'd be 4 for testing and 4 to dispense. That's the current 797. Moving on to the 797, that'll be official in November, says that, say it's that same 4 10 ml vials. So it says 10% of the batch rounded up, so 10% of 4 is 0.4. That's 1. So following the new rules, just need to make 5 vials, 1 for testing and 4 to dispense. As far as drug waste and things go, costs go, that's a big improvement for these really small batches. Just to drive home the point that no change has been made for batches greater than 40. Another example here would be for a batch size of 50 10 ml vials. Both 797 versions, one would follow Table 3 in 71, which in this case the 10% would be greater than 4, so 5 vials would be required for testing out of this batch size of 50. Something else to familiarize yourself with is that USP 71 has 2 tables, 2 main tables. Table 3 that we've just looked at in part is how to sample the batch, the lot, how to sample the lot. And Table 2 is how to sample the samples from the lot. So in this case, it was the 5 10 ml vials. So how to sample those 5 10 ml vials. The 10 ml vials falls in that 1 to 40 range. So it says test half the contents of each container, but not less than 1 ml. Important to note the title of this table is minimum quantity to be used for each medium. That's important because there are 2 media types. So when you're looking at that sampling in Table 2, keep in mind that there are 2 media types. The 2 media types are FTM or FTG and TSB. These 2 media types are designed to allow different types of microorganisms to grow so that they can be detected should they be present in the test samples. And to help promote the growth of those organisms, these 2 medias are incubated at different temperatures. So for a sterile result, both of these growth medias need to come back with no growth present. So I mentioned earlier about that USP said it had a preference for membrane filtration. I said that if I was a sterile compounder, I would have a preference for closed membrane filtration to be used in testing my drug products. And that's because closed membrane filtration is going to have the least likelihood of contamination to happen during testing, right? You don't want to go through all of that effort to make sure that you're producing a sterile compound preparation only to have contamination to happen during testing. The closed membrane filtration is the best apparatus for that. That is the apparatus here on the left. So just to quickly explain how this works, the drug product using a pump is pulled up through that needle. The tubing out of the needle then divides the drug product into 2 tubes that goes into those 2 canisters. Those canisters have a plug at the bottom of them. So the drug product essentially just runs through those canisters and that's a filter at the bottom of the canisters. The idea being that any contaminating microorganisms will be caught on those filters. You then plug those, plug the bottom of the canisters, and fill the two canisters with the two different medias using the pump. That's what's happening over on the right. And again, the two canisters get separated into the tube incubators. And then for the reeds, you need to look at both of them. So in comparison to this closed membrane filtration apparatus and open membrane filtration apparatus, think about it looking a lot more like almost like a coffee filter that would be sitting on top of one of these jars. So an open membrane filtration apparatus is really open to the test environment much more so than the closed membrane filtration apparatus. Something to keep in mind there, and just for reference, the direct inoculation would be one of these jars, just really being completely open to the test environment, and the drug product being added directly into those jars. So, something to take into consideration, and some things to ask about. So at the end of the incubation period, which is 14 to 18 days, the growth media is visually examined. So the material here, really how the results are obtained for both of these tests we talked about is, a visual examination. So mentioned here 14 to 18 days. That's because USP 71 has a mechanism built in. Should a test result be inconclusive at the end of that initial 14-day incubation period, you can incubate for an additional 4 days to determine whether that product is sterile or not, which is typically determined by turbidity in the media or visual growth. And so the 71 does have a fallback mechanism in case the test result is inconclusive. So 71 is always going to provide a test result, and if no evidence of microbial growth is found, the product meets the test requirements for sterility, and a sterile result is issued. So if a sterile result is issued, still really need to consider the chemical and physical stability for appropriate BUD assignment. If a non-sterile result is issued, then really need to have a full investigation on the compounding side and the testing side. So want to make sure that that investigation really needs to involve having an assessment, performing an assessment as to the state of the control in both the compounding and the test environments. And to be able to understand that, you need to have lots of data about what's happening in your environment. So the chapter has some requirements for how much EM is happening and air sampling and all those kind of things. Really need to think about whether that is an appropriate amount. That should you receive a non-sterile result, that you can perform a proper investigation, which you want to be able to do to identify the contaminating organism, because that can give you some insight as to how that organism entered the compounding space or the test environment and ultimately ends up in the sample. Okay, so something to consider there. Just briefly here on these alternative sterility test methods. So there are a variety of technologies and methods available. Many of them are designed to provide test results faster than 14 days, hence the term rapid sterility. But there is potential that most of these technologies are going to provide an inconclusive test result on a batch. So while there's lots of benefits, and the benefit being time, of these sterility test methods, there is potential that they're going to provide an inconclusive test result, which is not a possibility with the 71 test. So if you choose to use one of these alternative sterility test methods and receive an inconclusive result, that does not mean the product goes sterile. That does not mean the product goes non-sterile. It's just that it's an inconclusive test result. Because there's a variety of technologies available out there for rapid sterility, there's a variety of reasons why it may produce an inconclusive test result. So a couple of them are if the lab staff performing the test is unable to determine if the contaminant is microbial in nature. So some of the test results, the data is achieved by looking through a microscope or a computer monitor, trying to figure out whether something glowing on the filter that was used to filter the sample is microbial in nature. If they're unable to determine whether it's microbial in nature or not, that might be reported as non-sterile, that might be reported as inconclusive. Also, some of these alternative sterility test methods or rapid sterility test methods provide a signal. Well, the lab staff may not be able to determine if the signal is due to biological contamination. So if that's the case, that might be reported as non-sterile. That might also be reported as inconclusive. So just something to be aware of on these rapid sterility test methods. So in summary, USP 51 is a test to challenge the preservative's ability to kill or prevent proliferation in multi-dose, preserved, aqueous, sterile and non-sterile products. The 71 test ensures that the sterile product being tested is free from microbial contamination, but for appropriate BUD assignment, really need to consider the physical and chemical stability of these preparations. Here are some list of the references used throughout this presentation. Here is the evaluation link, which will also be emailed to you within about an hour or so. And I think we're going to open it up and see if there were any questions.
Thank you, Brian, for that great session. Reminder that you can still send questions using the questions tab, and we will get started.
Okay. So the first one says, if I already have stability data and didn't include a 51, should I perform a whole new stability study? Or can I just add a 51 to meet the chapter requirements? You could certainly just add that data. There's no requirements in the chapter that 51 be performed on the same lot at the same time or anything. As we talked about, really think about 51 as a formulation specific test or a formulation development type test. So it doesn't need to be performed at the exact same time as stability testing. Let's see. When should I implement the new USP 795 and 797 in my pharmacy? A good question. So USP does allow for for adopting those new chapters now that they're published official versions. Whether or not that's appropriate with your state board is something you should probably run past them. But as far as USP is concerned, those are the final versions, and so it's okay to go ahead and start using them. So will I have to perform method suitability on an alternative micro method if I already have method suitability for 71? The answer is yes. Different tests. Remember, the method suitability is a mini formulation-specific validation. Of the method, little mini validation of the method that's formulation-specific. So the method is changing from a traditional 71 to a rapid. You'd have to repeat method suitability. You said 51 should be done on all multi-dose vials, even if you're following USP BUD. I am specifically talking about trimix. Yeah, so that'd be a sterile injectable, likely aqueous. If that's the case, then the 797 does require 51 to be performed on that trimix. Remember, that's a formulation specific to the next question. Follow-up question was So no matter what your batch size is, now you have to test. Yes, but one time. That's a one-time. Remember, 51 is like a formulation development type test. That is not a batch release test. So it's a one-time activity. Remember, there are the allowances there for literature references on these trimixes. Also, probably a real good opportunity for the bracketing strategy to be implemented. Scroll down here. To confirm with non-sterile BUD assignment if ingredients such as Aura Plus or Aura Sweet, which already contain preservatives, would it be appropriate to assign a 35-day without USP 71 testing? So there's a few things about that one. That the Aura Sweet or Aura Plus, that's a non-sterile oral product, so 71 is not appropriate for those. So the 35-day BUD, maybe the intended question was 51. Okay, a couple things. If you're starting with a base such as Aura Plus or Aura Sweet, yeah, those are already going to contain preservatives. You need to consider what the concentration of the preservative is going to be after you compound, right? You're going to be adding ingredients in there that are likely not going to have a preservative in it, so you're thus lowering the concentration of the preservative in the final drug product. And so if you're going to assign a 35 BUD, you do not need to be concerned about 51, but you do still need to be concerned about the chemical and physical stability. Okay. Work my way through that. Can a pharmacy utilize the lab's method suitability library for 71 instead of doing their own method suitability testing? That's really a professional decision there. I think the science speaks well to doing that if it's the exact same formulation, as opposed to doing it on your formulation. So. Next question, where does oil-based ophthalmics fall under 51? 51 is intended for aqueous formulations only. Oil formulations are not appropriate, so so 51 is not an appropriate test. Again, think about that water activity test on your oil-based products to figure out whether 51 is even required. Are you saying that sterile products sent for sterility testing need to stay in quarantine until final results from the lab? Yes, according to the new chapter, need to wait until the final results are received but before those are administered to patients. Do I request a specific BUD for testing, or does the 51 test indicate the BUD based on extended testing intervals? No, so good question. So even though the 51 test has a bunch of time points, really the BUD is only supported by when that test was initiated. So if you really want a good data set, as I'm partially mentioned during the presentation, you'd want to initiate that. You might think about initiating the 51 at the end of the BUD that you want to assign, along with potentially a potency test on the preservative. That would make like a real nice data set for an extended test, extended BUD. The chapter doesn't really talk to that. It just says that you have to have it one time per formulation, but I would think putting it at the, initiating the 51 near the end of the BUD would be a real good design there. Can we not release any compound sterile preparation without the results? You can if you do if this is related to 71, if this question is related to 71, you can do it if it's released as a Category 1 and assigned a BUD per Category 1, or same for Category 2, but you'd have to follow the BUDs for drug products that were not subjected to a sterility test. If we already have a method suit on file, do we have to repeat the method suit for the new updated 797? No. Nothing has changed as far as that's concerned. So method suitability would still apply. Some non-sterile preparations have stability references, but not sterility. Can I assign a BUD based on my own sterility result, or should I send it outside? I'm not sure I understand that question, because it's some non-sterile preparations have stability reference, but not sterility. Amy, do you understand? You have to skip that one or ask for some more clarification. For CSP Category 3, sterility and endo are required despite the batch size. Sterility is required for Category 3 despite the batch size. I'd have to freshen up on the endotoxin requirements, but I think that has to do with the starting components to determine whether or not endotoxin is required for Category 3. So have you seen a batching process where the total volume of the batch is run through a filter, then that filter is tested? This would avoid wasting 10% for testing. So I think that's a sterility test question. As we talked about, the USP 797 has a preference for membrane filtration. We talked about that I would prefer closed membrane filtration versus open membrane filtration. All of the samples, as we looked at that example, like 5 vials would have to be tested. Those 5 vials are all ran through the same filter. It's not being separate filters, so there isn't any waste associated with it. Those were 10 mil vials, so 5 10 mil vials, all 50 mils of that is being ran through the 2 filters, and then the 2 filters are either collected during the open membrane filtration system, which I didn't really get into that a whole lot, but collecting the filter on the open membrane filtration system often requires the use of tweezers and scissors. You have to take that device apart, whereas on the closed membrane filtration, the media is just added directly into those canisters. So there's really not any waste. I mean, the drug product does run right through the filters into the waste, but the idea that any contaminating microorganisms would be caught on those filters. For 795, do you have to test 51 if you don't want to extend the BUD? No. The 51 is only required for multi-dose aqueous if you want to go past the 35-day. So what does bracketing for 51 testing look like for a preserved product with multiple APIs and it offered at different concentrations for those APIs? So that's really where you get into what's referred to as matrixing as opposed to bracketing. Bracketing is sort of a simple version for formulations mostly intended with a single API, but there is something such as matrixing. I would think that if bracketing is allowed, then the appropriately designed matrix would also be scientifically justifiable. It's just a more complicated process than a simple bracketing strategy, but can be done. Would stability indicating studies for any CSP include 51? That could certainly be part of a stability study. As I mentioned, it'd be nice to have it at the end. That's not a requirement in the chapter. The requirement in the chapter is just one time per formulation per container closure system. So a 51 could certainly be performed outside of the stability study, and you could just package it up, that data with your stability study data. once all that testing was performed. For non-sterile preserved aqueous dosage form, is the preserved defined by generally accepted preservative quantities instead of a 51 test? So I think the question there basically is a potency test, or just knowing that my preservative is at, is at kind of a standard level. Is that good enough? And the answer is no. Talked about that there's a lot of opportunities for interactions between the preservative and something in the formulation, the preservative in the container closure system. The preservative could be at the wrong pH, for example. So all of those things could minimize the effectiveness of the preservative, and so that's the purpose of the test. Will you have this presentation recorded? Yes, you will receive a copy of that within about an hour. Okay, a couple more. Is the appropriate way to bracket a study for 51 to test your lowest and highest concentrations containing the appropriate corresponding amount of antimicrobial? So for appropriate bracketing per the chapter, they should all contain the same amount of preservative. The only difference would be the amount of API, active pharmaceutical ingredient. If that's the case, then yes, you would test the highest and lowest concentrations. Any concentrations of the API in between could be covered by that bracketing. So if benzyl alcohol is used at 0.9% for CSP, but pH is maintained at 6.5, more than recommended, can we still proceed with 51, although it's out of its recommended range, or what should be the right course of action could be? So those were optimal pHs. There's certainly opportunities to still pass a 51 test when you're outside of the optimal pH. So could run the test to see if it passes, or could see if there's a way to change that pH so that it is closer to the optimal pH for the benzyl alcohol, so that you have a higher level of confidence the test is going to pass. Okay, I think we covered the majority of the questions there. So.
Yeah, great. Thank you guys so much for the excellent questions and the great Q&A session. Thank you, Brian. As a reminder, a recording of this presentation, the link to the slides, and the evaluation link will be sent in a follow-up email 1 hour after today's live session. And we appreciate it, and have a great day.